Okay, let's unpack this, because today we are looking at something that honestly feels like it was ripped straight out of a science fiction novel.
Oh, absolutely.
But it is happening right now in real life in the world of computational biology. We are exploring how artificial intelligence is teaming up with algorithms inspired by wild animals.
Yeah, and the laws of nature.
Right. To solve one of the most massive biological puzzles of our time, which is mapping the human genome. And for you listening, we've got a stack of notes here based on some incredibly dense, highly technical excerpts from a 2026 CRC press book.
It is remarkably dense.
Yeah, it's titled Deep Learning in Genome Mapping, Computation, and Analysis. Specifically, we're focusing on chapters 1 and 2 by Shailendra Pratap Singh.
It's a phenomenal, though, very heavy text.
Very heavy.
And our mission today is to take these deeply complex computational theories, the kind of math that usually stays locked inside university supercomputers, and translate them into a digestible, fascinating story about the future of human health. We're going to see how mimicking the behavior of humpback whales and even the fundamental laws of evolution itself is currently helping computers read our DNA faster and, well, far more accurately than we ever thought possible.
I just love that mental image. A supercomputer wearing a tiny captain's hat, hunting for data like a whale.
It's a great visual.
But before we get to the marine biology part of the AI, we really have to understand the sheer mind-boggling scale of the problem. Genome mapping is essentially the process of determining the precise locations of genes and other vital markers within an organism's DNA.
Right.
If you want personalized medicine or evolutionary biology or, real-time disease prediction, you need an accurate map. Now, back in the day, mapping relied on older algorithms. You might be familiar with BLAST.
Right, the basic local alignment search tool.
Yeah, that's the one.
And BLAST was absolutely revolutionary back in the 1990s for sequence alignment. It gave scientists a way to actually compare biological sequences. But today, we've moved into the era of modern high-throughput sequencing technologies.
Which changes everything.
Completely. These machines are churning out genomic data at a rate that is frankly overwhelming. We've created a massive data bottleneck. There is simply too much complex genetic data for traditional heuristic-based methods to handle efficiently.
And by heuristic, we basically mean those older rule-of-thumb algorithms that make educated guesses, right?
Exactly. They just can't keep up with the hidden dimensions of modern data.
So we hit a wall with the old tools, the data got too big too fast.
What's fascinating here is how the scientific community pivoted to address that bottleneck. They realized that deep learning, specifically convolutional neural networks, or CNNs, and recurrent neural networks, RNNs, offered a really powerful way out.
Because they can see things we can't.
Right. Deep learning models are incredible at capturing hidden spatial hierarchies. They can recognize highly intricate patterns in raw genomic data without needing a human to manually label every single feature for them. But, and this is a massive, but deep learning requires an enormous amount of computational power.
Oh, yeah.
You can't just throw raw computing power at this problem and expect it to scale indefinitely.
Because eventually you just run out of servers.
Or electricity.
Or time, yeah.
Precisely. The core thesis we are looking at today is that the ultimate solution isn't just building bigger servers. It's combining those deep learning architectures with highly intelligent, nature-inspired optimization algorithms.
So we've got to make the software smarter, not just the hardware bigger.
Exactly.
And that leads us to a specific type of deep learning architecture discussed in the text called graph neural networks, or GNNs. To make this relatable for you listening, I want you to imagine your genome not as a twisted ladder or a straight line of code like you see in the textbooks.
Right, forget the straight line.
I want you to imagine it as a massive, bustling social network. Imagine every gene in your body is a person on this platform.
I really like that analogy. In a graph neural network, we define the data mathematically as a graph. You have nodes, which represent the genes or the specific genomic regions.
The people in the network.
Yeah, right. Then you have the edges, which are the connections between those nodes. Those edges represent the relationships or interactions, things like gene co-expression, shared mutations, or regulatory interactions.
How they talk to each other.
Yes. Traditional linear mapping tools look at a DNA sequence, like a long, straight string of letters. But biology isn't linear. It's an incredibly complex web. At each layer of a GNN, the representation of a specific node is updated by aggregating information from all of its neighboring nodes.
So just like in a social network, you are defined not just by your own profile, but by who you interact with.
Exactly.
The network updates a gene's profile by looking at what its friends are doing. But how does the computer keep all that straight without the popular genes completely drowning out the quiet ones? Because the text mentions some pretty heavy math behind this.
It does get mathematically intense. The researchers use something based on a normalized adjacency matrix, but if we translate that out of the textbook jargon, the math essentially acts like a sophisticated filter. It ensures that a highly connected, popular gene doesn't completely drown out the voices of quieter, but equally important neighboring genes. The model learns real representations that capture both the individual properties of the genes and their broader structural neighborhood.
That makes sense.
This graph-based approach allows researchers to capture hidden relationships that traditional straight-line mapping completely misses. It's a total paradigm shift in how computers see biological data.
Yeah, and they actually put this to the test on real medical data, right? It was a diabetes data set.
They did. And the sheer size of it is staggering. We're talking about mapping nearly 20,000 individual nodes, 19,717 to be exact, with over 44,000 connections between them.
Wow.
Processing that much relational data is mathematically exhausting.
Right. So we built this massive genetic social network to solve the brain power issue, but that creates a new problem. Speed and computing exhaustion. Because if the model takes a month to compute, it's not very helpful for a patient waiting for test results. Here's where it gets really interesting. How do we make something that smart run fast enough to actually be useful?
Well, the deep learning model needs a co-pilot. The neural networks are brilliant at pattern recognition, but configuring their internal weights, essentially fine-tuning the model so it makes accurate predictions quickly, is a major hurdle.
It takes too long to train it.
Way too long. So to solve this, the researchers brought in something called the Differential Evolution Optimization Algorithm, or DE.
So if the graph neural network is the brain recognizing the patterns, the DE algorithm is the evolutionary pressure forcing that brain to get smarter and faster.
That's a great way to put it. Differential evolution is a brilliant concept. It essentially mimics biological survival of the fittest to mathematically optimize the deep learning model's parameters.
Okay, break that down for us.
The text lays out 3 specific steps of this algorithm. First, you have population initialization. The algorithm starts with a massive random pool of candidate solutions, essentially, different sets of mathematical guesses for the neural network.
A whole generation of slightly different algorithms just thrown at the wall to see what sticks.
Yes. And then comes the second step, mutation and crossover. The algorithm mathematically combines existing solutions to explore entirely new possibilities.
Like breeding them.
Exactly. It takes 3 randomly selected individuals from the population, mutates them together using specific scaling factors, and crosses them over with the current candidate to generate a trial solution. You are basically breeding the mathematical guesses together to see if their offspring is smarter.
And then comes the final ruthless step, selection.
It is entirely ruthless. The algorithm tests that trial solution's fitness. Now, instead of getting bogged down in the specific cross-entropy loss functions mentioned in the text, just think of it as a rigorous mathematical test of accuracy.
A pass-fail grade.
Right. If the new mutated solution is more accurate at mapping the genome than its parent, it survives to the next round. If not, it's deleted. Only the absolute fittest candidates survive generation after generation.
So they ran this head-to-head against other models. What happens when you force code to mutate and survive over hundreds of generations?
The results are staggering. The researchers ran this proposed DE-BAG algorithm over 300 generations, and they pitted it against some of the most established state-of-the-art optimization models in the industry.
Like the baselines.
Right. Heavy hitters that are normally the gold standard for this kind of math, like traditional genetic algorithms or GA, particle swarm optimization, PSO, and the Gray Wolf optimizer, GWO.
And how did it do?
The proposed algorithm completely smoked the competition.
Give us a sense of the scale here. How much better are we talking?
Well, instead of getting stuck at a B+ like the older models, this evolutionary approach pushed the accuracy to a solid A. It achieved a best accuracy of 95.24% and a mean accuracy of 90.50%.
That is massive.
It really is. In the world of genomics, that 5% or 10% difference could mean the difference between catching a genetic disease marker and missing it entirely.
And a big reason it performs so well, according to the text, is that it avoided something called premature convergence. Can we unpack that?
Yeah.
Because it sounds like a bad date, but it's actually a major coding problem.
It's a huge problem in machine learning. Premature convergence happens when an algorithm gets trapped in what we call a local minimum. Basically, it finds an answer that is good enough, assumes it has found the best possible answer, and completely stops looking.
It just packs it in.
Right. But because the differential evolution algorithm maintains such a diverse population through its mutation and crossover steps, it continuously explores the vast search space of the genomic data without getting stuck.
It keeps looking.
It balances looking for new answers with refining the good answers it already has. It offers a much more stable, robust learning curve than anything that came before it.
So we have accuracy. We have an algorithm that acts like a constantly evolving species to get us a solid A in reading DNA. But genome mapping isn't just about pure accuracy, is it? It's what the text calls a multi-objective problem.
That's the real challenge. In the real world, particularly in clinical settings, you want your genome map to be perfectly accurate, yes, but you also desperately need it to be incredibly fast.
Right.
Those are two conflicting objectives. Usually, if you want a highly accurate computation, it takes a massive amount of time. If you want it fast, you have to sacrifice precision.
It's the classic, you can have it fast or you can have it right, but you can't have both dilemmas.
If we connect this to the bigger picture, what researchers are looking for is called a Pareto optimal solution.
Pareto optimal.
Yes, it's the mathematical sweet spot. It is the perfect, uncompromisable balance between computing time and mapping precision, where you cannot improve one objective without directly hurting the other.
And to find that elusive Pareto optimal solution, the text introduces a totally different, visually stunning concept in Chapter 2. The multi-objective whale evolutionary algorithm, MOWEA.
It is beautiful biomimicry. MOWEA is mathematically structured to mimic the highly complex social behavior and hunting strategies of humpback whales.
Which is wild to think about inside a computer.
It is. Humpback whales are incredibly intelligent pack hunters, and their strategy maps perfectly onto the problem of searching a massive, complicated data set.
I really want to visualize this for everyone listening. The text breaks down the whale's behavior into two main phases that translate directly into the algorithm's math. First, you have exploration.
Imagine the whales swimming through the vast open ocean. They are searching randomly for prey. In the algorithm, the computer is doing exactly that. It's searching the massive dimensional space of the human genome randomly, looking for promising genetic sequences or patterns.
And then comes exploitation. This is my absolute favorite part. When humpback whales find a school of krill or small fish, they don't just swim into it blindly.
Right, they have a system.
They use social interactions and a highly coordinated technique called bubble net feeding. They swim in shrinking circles around the prey, blowing bubbles from their blowholes to trap the fish in a tighter and tighter cylinder of air.
It's a highly sophisticated trap. And in the algorithm, once a promising genetic mapping solution is found, the math shifts. The algorithm mathematically updates the position of its whales, its candidate solutions, by drawing them closer and closer to the best known solution.
And the source explains exactly how the algorithm pulls off this balancing act between exploring the ocean and blowing the bubble net. It uses specific mathematical coefficients that adjust dynamically over time.
Yes.
Now, we don't need to get bogged down in the formulas, but basically there's a mathematical variable that dictates the encircling behavior. It decreases steadily over the course of the computations, effectively tightening the search radius, just like the whales tightening their bubble net around the target.
It's very similar, though mathematically it's actually a bit more rigorous. It forces the computer to constantly balance randomly searching new areas of the data with aggressively refining the best answer it has already found.
Right.
And because it's a multi-objective algorithm, it is constantly measuring every movement against that Pareto optimal sweet spot we mentioned. It asks itself, is this new mathematical position both faster and more accurate? If yes, it becomes the new best solution.
And the proof is absolutely in the data here too. The researchers subjected this whale algorithm to an incredibly rigorous testing ground. They ran it over 600 generations.
Double the time of the previous tests?
Yeah. And they tested it against a whole suite of state-of-the-art algorithms that are normally the absolute top tier for these kinds of multi-objective problems. Things like NSGA2, MOPSO, and MODE.
Not only that, but the data sets they chose to test it on were very deliberate and highly complex. They used a full human genome data set, commonly referred to as the HG data set, which is the gold standard benchmark.
Right.
They used the Arabidopsis data set, which is a fundamental reference data set for plant genome research. And finally, they used a GWAS data set.
Let's unpack GWAS for a second, because that's a term that gets thrown around in biology a lot. It stands for Genome-Wide Association Studies.
Yes, and it is notoriously difficult for computers to process. A GWAS dataset is basically looking for the needle in the haystack. It's trying to link a tiny, specific genetic variant to a big, complex physical trait, like a disease symptom or a physical characteristic.
It's looking for those tiny connections.
It requires looking at millions of genetic variations across many individuals to find those subtle connections.
So they threw the hardest possible tests at this whale algorithm. How did it do?
It dominated across the board. After 600 generations on the human genome dataset, MO-WIA hit an accuracy that translates to a solid a 0.94 accuracy. On the plant dataset, the Arabidopsis one, it hit 0.92. And even on that wildly complex G2SS dataset, it achieved a 0.90 accuracy.
So what does this all mean? We've talked about deep learning, graph networks, mutating algorithms, and bubble netting whales. Why should you, the listener, care about the fact that an AI is acting like a whale to read human DNA?
Because this isn't just an abstract mathematical exercise happening in a vacuum. This is the bedrock of precision medicine.
Yeah.
The sheer volume of genomic data we produce today holds the answers to some of the most devastating diseases on the planet. But those answers are hidden in a sea of noise. The text specifically highlights how accelerating this mapping process directly impacts disease prediction.
Real-world applications.
Exactly. It specifically mentions applications in identifying lung cancer, brain tumor classifications, and dramatically accelerating drug discovery.
So when a doctor sequences your DNA in the future to figure out if you're at risk for a specific cancer, they won't have to wait months for a supercomputer to chug through the data.
No, they won't.
Because of these whale-inspired algorithms, they can get highly accurate results fast enough to actually act on them. The text gives us a really cool visual representation of how this data is ultimately used in the real world, the heat map of gene expression correlations.
The heat map is a critical output of these mapping analyses. The source text describes a visualization showing the pairwise correlation coefficients between unique genes across all samples.
And it's color-coded.
Yes, it uses a color gradient. Red indicates strong positive correlations, while blue indicates negative correlations.
So when a researcher sees bright red spots on this map, they are looking at co-expressed genes that are essentially turning on and off together.
Exactly. And discovering those co-expressed genes is like finding a brand new biological pathway. It helps scientists discover hidden biomarkers,
Which is the key to treating the disease.
It tells doctors not just what your individual genes are, but how they are actively talking to each other, how they're regulating one another, and how they might be teaming up to cause a disease. It allows us to identify entirely new therapeutic targets for drug developers. Instead of attacking a disease blindly, we can see exactly how the disease communicates on a genetic level.
It's genuinely incredible. To wrap up this journey we've been on today, we are looking at a future where the sheer, overwhelming complexity of human biology is being decoded by artificial intelligence. But the AI wasn't smart enough or fast enough on its own.
No, it needed help.
To push the AI forward, computer scientists had to look backward. They had to look at the laws of nature. They mathematically coded the evolution of species and the survival of the fittest. They coded the social interactions and pack hunting strategies of humpback whales.
It's amazing.
They used the brilliance of the natural world to optimize the brilliance of the artificial world.
This raises an important question, and it's something deeply profound to think about. If human intelligence is now utilizing the mechanics of biological evolution and the complex behaviors of wild animals to teach artificial intelligence how to decode our own human biology,
Right.
Are we just closing a massive planetary feedback loop? Could it be that the ultimate key to understanding our own biological source code isn't going to be found in a sterile, isolated lab, but by looking much closer at the intricate, ancient, natural ecosystems that are already thriving all around us?
That is a wild thought to leave off on. We are basically using nature to teach computers how to read nature. A massive thank you to you, our listener, for joining us on this deep dive into the fascinating intersection of wild ecosystems, AI, and the genome. Keep questioning, keep exploring, and we will see you next time.